Hi everyone, this is Dr. Omar. In today's presentation, I will describe a key threshold concept in molecular biology, that is Watson and Crick DNA model. And within this presentation, I will explain the significance of this threshold concept in molecular biology towards the end of the presentation. But basically I will go through the Watson and Crick model DNA first. Nucleic acid is the molecules of life. It stores and transmits genetic information. Two types of nucleic acid: DNA, deoxyribonucleic acid; RNA, ribonucleic acid. The nucleic acid itself consists of long polymer of repeating units called nucleotides or deoxynucleotide, whether it is RNA or DNA. The nucleotide itself is composed of three parts or components: pentose sugar, as shown by here in the diagram, phosphate group, and nitrogenous bases or base. The pentose sugar has 5 carbon atoms in its structures, and they are numbered from 1 to 5. RNA contains ribose at carbon number 2, and the ribose has OH group at carbon number 2 in its attached to it. The DNA contains deoxyribose, and at the carbon number 2 an H atom is attached instead of OH, so it is deoxy. The oxygen is removed, the oxygen atom is removed, so it is you have two types of sugar, ribose and deoxyribose. The nucleotide also contain nitrogenous bases. There are 5 different nitrogenous bases in the nucleic acid, and they are divided into 2 categories: purines, double ring structure, that includes in it adenine and guanine; pyrimidine, single ring structure, that includes thymine, cytosine, and uracil. In the next slide, we see here a diagram of the essential component of the nucleotide structure: phosphate group, sugars, 2 types, deoxyribose and ribose, and different nitrogenous bases depending on whether the molecule is DNA or RNA. Now, as we can see here in this diagram, you have the nitrogenous base, sugar molecules, and phosphate. When the base is attached to a sugar without phosphate, it's called nucleoside. When the phosphate is attached, it's called nucleotide. So that's the main difference between nucleotide and nucleoside. In nucleotide, the presence of phosphate group will make the molecule named nucleotide, and without phosphate it is called nucleoside. Now, how are these nucleotides joined together to form a polymer or a chain or a single strand of DNA? Through a phosphodiester bond. This phosphodiester bond is formed between phosphate group in one nucleotide and sugar molecules in the adjacent nucleotide. Phosphodiester bond involves phosphate attachment between the 5 group in one nucleotide and the 3 group in the 3 carbon in the another. So it between 5 carbon in one nucleotide and the 3 carbon in another nucleotide. So the single-stranded DNA, the nucleotide are linked together in a linear manner, as it's shown here in this diagram, to form DNA strand. Phosphate group and the sugar molecules form the backbone for the DNA strand. And as we can see, the nitrogenous bases protrude or project from the backbone. And the orientation of this nucleotide is determined by the orientation of the sugar. So the strand can be from 5 to 3 or 3 to 5 based on the orientation of the sugar. Now this is another example diagram for the DNA, short DNA strand. So you can see here the direction of the strand is from 5 to 3. Strand contains a specific sequence of bases. The sequence of the bases are thymine, adenine, cytosine, and guanine. The way to write that is 5 terminus, 5 end, TACG, and then 3 hyphen, which is 3 terminus, 3 end. Now Watson and Crick has postulated a model for how the DNA looks like and how it is joined together, two strands, to make the double-stranded DNA. They postulated that the two DNA strands twist together around central axis to form double helix, double-strand DNA, as we can see in the small diagram here. The two DNA strands are anti-parallel. One's in the 5 to 3, the other is from 3 to 5, so they are opposite orientation. The double-stranded chain are stabilized by hydrogen bond between opposite bases. Each strand of a helix is complementary to the other, so there is bearing between one strand and the other strand, and the nitrogenous bases between in each strand bear with complementary nitrogenous base in the other strand or opposite strand. Adenine base is one strand, forms 2 hydrogen bonds with thymine base on the opposite strand. Guanine form 3 hydrogen bonds with cytosine, and we know that adenine bear with thymine and guanine pairs with cytosine. Again, as we can see here from the diagram, it's a double helix, 2 strands of DNA, opposite direction, orientation, in between the nitrogenous bases. These nitrogenous bases are stabilized and bind to each other through hydrogen bonds, 2 bonds or 3 bonds depending on the nitrogenous base type. The DNA sequence have a high proportion of GC content, tend to form more stable double-strand structure, as we can see here from short oligo or short fragments of double-stranded DNA. The model that's suggested by Watson, postulated and hypothesized by Watson and Crick model, is right-handed model. It has 2 grooves, the minor grooves and the major grooves. Now, why do we think that this is a significant and important threshold concept in molecular biology? Because the DNA model, double-stranded DNA, the structure of the DNA is forming the foundation for comprehensive understanding the disciplines. Once mastered, it will transform the student's subsequent perception and cognition of the subject. Once this TC concepted, it will facilitate apprehension of further related central ideas based on this concept. It will permit constructing coherent but sequential and organized recognition of successive principles and knowledge. The threshold concept will allow integrating further acquired information based on the TC and will enable reformulating coherent overall view of the subject. The threshold concept will enable and broadening venues of lateral thinking and understanding of complex facts and solving factual challenged queries, thus reaching utmost mastery of the discipline. Once students perceive the threshold concept, they will be immersed in all scopes of discipline directly or indirectly. It will be the stem. The threshold will be the stem for intercalating, overlapping, or bridging ideas, facts within a subject or between related disciplines. The threshold concept is the gateway to overcome learning difficulties and complex principles and information. And these are the most crucial explanation of why this threshold concept is important in molecular biology. Now, to address it furthermore, understanding and grasping and mastering the concept of DNA model, double-stranded DNA, the structure of the double-stranded DNA, how the two strands are aligned anti-parallel to each other, and how they are bound to each other through hydrogen bond, form the foundation for understanding a process which is called replication. It's an in vivo DNA synthesis and copying. It's essentially based on understanding the structure of the DNA, double-stranded DNA. Again, another topic that is really crucially based on better full understanding of the DNA model is a technique that is used to analyze DNA molecules, and this is quantitative real-time PCR or PCR itself or digital PCR. It's an in vitro DNA synthesis and copying. And also understanding the DNA model is important for understanding the central dogma, as the central dogma is copying of the double-stranded DNA and then expressing of the gene. So understanding that DNA model will facilitate better understanding the structure of the gene. Again, also a technique that is really crucial in molecular biology for analysis of molecules of DNA looking for variation within the sequence is called DNA sequences, and it's involved in vitro DNA synthesis and copying. Again, having a strong and enough and grasp background of the structure of the DNA is essential to understand these methods. Recombinant DNA technology, where a DNA piece of double-stranded DNA cloned into another carrier of DNA, is also involved in vivo DNA replication, in which copies and clones of the target DNA is allowed to increase in number and produce clones of specific the fragment of DNA. So it's essential to understand the DNA models, and also DNA model structure is important to understand the gene structure. Thank you very much for listening.